Hey, AP Bio. All right, here we go. Some notes for this gene regulation stuff. So these are pretty short notes, but I'm still going to split it in half just to keep it short, okay? So we are focusing on prokaryotes today and how they control the expression of their genes. All right, so a little bit on bacteria and bacterial metabolism. So bacteria really live in their environment, right? I mean, that doesn't really make sense, but we are fairly sealed off with our chemical processes in our body, right? We maintain a pretty steady homeostasis and are not influenced by the molecules floating around us as much. Okay, bacteria on the other hand, what is around them is what they are going to use for their metabolism and growth and reproduction. And so, if all of a sudden, for example, you drank a big glass of milk, ew, gross. um, your gut bacteria would all of a sudden be overcome by all this lactose. And if they can break down that lactose for food, they're in good shape. And if they couldn't, they would lose all the energy from that molecule. So they would want to be able to quickly turn on the genes that regulate the production for the enzymes that break down lactose, so they can do that really quickly. And if they don't do it fast, then boom, that lactose is gone and they didn't get the energy from it. So they need to be able to respond to their environment incredibly quickly. And so gene regulation and being able to turn on and off separate genes quickly is very important to them. And so even though a lot of it is similar in eukaryotes, there's a few things that are different in prokaryotes because they are trying to respond so quickly to their environment. All right. So there's two general types of ways they regulate genes, and that is if they have enough of something, like this is usually a gene that's continually on, but then if they stockpile enough of something, they need to shut that off so they're not wasting energy. And then the one that I was kind of referring to with lactose was all of a sudden they're exposed to something that they need to respond to or use, then they might need to turn on genes for particular enzymes to take advantage of that situation. Okay. And so those are the two that we're going to focus on: repressible genes and inducible genes. And we're going to use tryptophan and lactose as an example of those, and that'll probably be in the second half. All right. So we already mentioned a long time ago, first semester, that we can use feedback inhibition to regulate processes, regulate metabolic processes, chemical processes. So, and the tryptophan was the one that we used in our examples there. So we make enough tryptophan. I think that comes from isoleucine. And so we've gone through this metabolic pathway. We use a series of enzymes to change isoleucine into tryptophan. And when we get enough that we have plenty of tryptophan, it will actually go back and be an allosteric inhibitor. So remember, that's a molecule that bonds to another site on the enzyme that ends up shifting that active site so it no longer fits to the substrate. However, I mean, that's a good quick fix. So it's a good quick response. However, you're still using those genes and going through all the processes to make those enzymes for that metabolic pathway. And so that's kind of wasting a bit of energy. So gene regulation is a better way to regulate because you're not going to end up wasting all that energy building enzymes that you don't need. Okay. Oh, good, Mr. Penguin. You remembered. Awesome. All right. So gene regulation, like I said, we're wasting a lot less energy because we're going back to the DNA itself, blocking the transcription of that DNA so that we don't waste energy going through protein synthesis and making all these enzymes. All right. So how does that work is what we're going to be talking about in here. We can regulate genes by turning on transcription and allowing transcription to occur, or turning off transcription. Okay, and like I mentioned earlier, we're going to use tryptophan and lactose as examples of that. Okay, so a repressible example is tryptophan. We build a bunch of it, we have excess, we go back and turn off those genes. Lactose is an example of inducible. Lactose becomes present, okay, so it's going to turn on those genes to make those enzymes. The stop and go, repressible, inducible. All right, so we need to understand a little bit more about just how bacteria organizes its genome, okay? Bacteria, very clever, uses this operon technique, okay? And it's where it lumps a bunch of genes that are all related to the metabolic pathway together. It is a fairly unique prokaryotic way to organize genomes. There are limited eukaryotic operon examples, and they're usually in lower life forms, like a nematode, I think, has a few. Okay, but for the most part, they're a prokaryotic phenomena. Okay, and so what it has is some of these things should sound familiar. It has a promoter region before the gene, right? You know all about this. So this is the portion of DNA before the gene of interest. It will have the TATA box in it. It is where RNA polymerase attaches. You know this, okay? But something we have not mentioned, which is in the promoter region as well in an operon, is the operator. Okay, and this is where the repressor protein can attach. And actually, we do have repressor proteins in eukaryotes as well. Okay, so repressor proteins can attach in the promoter and stop the ability for that RNA polymerase to move down the gene of interest. Okay, so it stops it, it blocks it, and then all those genes afterwards are not going to be transcribed. And this is what's very unique about prokaryotes, is you can see there it has 5 genes all regulated or that all are used in the metabolic pathway to produce tryptophan. Okay, they're like E, D, C, B, and A. So it actually makes an mRNA sequence, and remember, we don't have introns in prokaryotes. It makes this mRNA sequence that the ribosomes can attach to and like make all 5 enzymes right there. Boom, boom, boom. Okay? Very clever. And so as it goes through there, it reads AUG. It starts trip E transcription. It reads a stop, so it releases that enzyme. Then it continues down the script. It finds another start for trip D, and so forth. Okay, and so an operon is the grouping together of all those enzymes in the same metabolic pathway. Okay, so we have in the promoter region the usual like TATA box, etc., but we also have that operator where the repressor protein can bond, and then we have the operon itself. The majority of the operon where it has multiple genes for the same metabolic pathway. Okay. In that, on the operator, the repressor molecule can attach, and it's going to be like semi like a lock and key, like they fit together. Okay. And so if that repressor protein fits, it will attach to the operator and it'll stop the RNA polymerase from continuing through transcription. Okay. It can be that if there's an excess of something like the tryptophan, there'll be more bumping around. So it bumps into that repressor protein, changes the shape so that it fits to the operator. The opposite would be true of lactose or the lac operon. It would be stuck there And then in the presence of lactose, it would attach to the repressor molecule, change the shape, and it would release from the operator. And that's what we're going to look at two more examples those two examples in a little bit more detail tomorrow. Okay, so this is the end of the notes for today. Just a little bit on the repressor molecule. You can see that it fits to the operator and stops transcription, and RNA polymerase being able to do that. All right, so that's our little bit on gene regulation today, stepping into it and the idea of an operon. And then tomorrow we'll look at specific examples in a little more detail. All right, awesome. Have a great day. Thanks, guys.